Welcome to Quantum Photonics. We are here with this very, very interesting topic, and actually, it's very helpful for people who are also for health, and of course, people who are very conscious on how they look. But I myself, I would like to say that I have a sweet tooth, and I'm very interested to listen to what our speaker has to say. So, people who find themselves rummaging around in the refrigerator for a snack not long after they've eaten a filling meal might have overactive food seeking neurons, not an overactive appetite. UCLA psychologists have discovered a circuit in the brain of mice that makes them crave food and seek it out even when they're not hungry. When stimulated, this cluster of cells propels mice to forage vigorously and to prefer fatty and pleasurable foods like chocolate over healthier foods like carrots. This is the paper that we will be discussing, the paper of our guest speaker here, whom I'd like to introduce to you. Dr. Abhishek Adhikari joined UCLA Psychology Department in 2016 following a postdoc training at Stanford University with Professor Carl Diceroth and PhD studies at Columbia University with Professor Joshua Gordon and Professor Rene Hent. Dr. Adhikari's lab investigates how the brain coordinates the constellation of changes related to emotional behaviors with a focus on fear and anxiety. And these multifaceted changes involve a complex and dynamic adaptations in hormonal, physiological, and behavioral realms. Dr. Adhikari dissects how interactions between brain structures control these processes, seeking insights that shed light on the neural basis of pathological anxiety disorders and adaptive aversion to danger. To do so, they use a combination of powerful techniques including electrophysiology, behavioral assays, optogenetics, and calcium imaging to monitor and control neural activity and behavior. So, I will try to link his lab website here at the room chat later on. But for now, I am excitedly trying to introduce him to you. So I bring you Dr. Avi Adhikari. Avi, welcome to Quantum Photonics.
Thank you, Cecile, for inviting me, and I'm very happy to share our newest work where we discovered a circuit in the mouse brain that controls food seeking even if the mouse is not hungry. So I would like to ask everyone to first just take a moment to open the PDF of the presentation slides that's linked to this presentation in the room. So please open the PDF first. Okay, so now I'll say which slide I'm on in the PDF every time I advance. Okay, so first in slide 1 of the PDF, we have this picture of a mouse eating a cheese, which is the behavior we're studying, which is food seeking. And these results that I'll talk about have been published in this journal, Nature Communications, and the link for the journal article is there on slide 1. Okay, so now let's go on to slide 2. So here in the next few slides, I will first explain how the optogenetic method works, which is the method I used for most of the data I'll show today. Okay, so optogenetics is a method to selectively activate or inhibit different types of neurons within a brain region. So for example, neuroscientists might be interested in knowing what do these particular neurons in this nucleus of the brain do to control sleeping or eating or something like that. And then they can use optogenetics to inhibit or excite those cells and then see what happens in the behavior that they're interested in. So this is the method that was used for most of the data that I'll talk about next. So first, a quick background so it's everyone can follow how the method works. And as you have already heard from previous places, neurons are like electrically active cells. And when they become electrically active, they release neurotransmitters onto other neurons. And that in turn alters the electrical activity of other neurons. So neurons become electrically more active when positive charges go inside the cell. And if enough positive charges go inside the cell, the neuron will create a very fast electrical impulse and then will release some neurotransmitter. And that neurotransmitter could be something like serotonin or dopamine, which are famous neurotransmitters. But the neurotransmitter that we studied in this investigation is called GABA. And GABA is a very important inhibitory neurotransmitter, meaning that when this transmitter is released onto neurons, those neurons become electrically more silent. So we use this optogenetic method to activate or inhibit these GABAergic cells in the midbrain of the mouse. Okay, so now how does optogenetics work? So it's a method to use light to excite or inhibit electrically cells. So normally our neurons inside our brain are not sensitive to light because there is no light inside the brain, so it makes sense that the neurons don't have any mechanisms to detect or respond to light inside the brain. But there are several unicellular organisms that have been found that have some sorts of proteins in their outer membrane that are sensitive to light. And then in the presence of lights of particular colors, those proteins are activated and then they can induce a influx or outflux of positive charges from the cell. So here, if you look at the figure on the right, you see an example of one of these proteins. So the left panel of the figure, you see this CHR2 written, which is an abbreviation for channel rhodopsin. So this is a protein found in a microorganism that, in the presence of blue light, lets in positive charges like sodium inside the cell. So if you put blue light on this protein, more positive charges go inside the cell, and the cell becomes potentially electrically activated. And in another microorganism, it was found this other protein that we see on the right called ARCH. And in the presence of green light, ARCH pushes out positive charges, represented here by this positive hydrogen charge. Uh, so the CHR2 can be used to electrically activate neurons because it lets in positive charges inside the neuron, and the ARCH is used to electrically inhibit neurons because it pushes out positive charges. And then remember, the more positive charges accumulate inside the neuron, the more electrically active it becomes and the more kind of neurotransmitter it releases. But these are, as I mentioned, proteins that are found in unicellular microorganisms, so they're not found in the brain normally. So we used artificially engineered viruses, and those viruses have inside of them the DNA to make either this excitatory channel rhodopsin or this inhibitory ARCH protein. Then we injected these artificial viruses inside the mouse brain. Then those viruses infected the cells we wanted to study, which are the GABA neurotransmitter releasing cells in the midbrain. And we can genetically engineer the virus so that it selectively only affects one particular type of cell in the brain. So even within a very tiny brain region, there might be many, many different types of cells. So within one little nucleus, there could be cells that use serotonin as a neurotransmitter, or dopamine, or GABA, or acetylcholine, or some other neurotransmitter. And often you'd want to study not how that whole brain region participates in a behavior, but selectively how do the, say, serotonin releasing cells in that region control a behavior. So this virus can be engineered to infect only cells of particular type in that brain region. So the channel rhodopsin of the or the arch would be produced only in one particular cell type. So in this case, we're studying the GABA neurotransmitter releasing cells in the midbrain region of the mouse. So we injected this virus there, and then the virus infected those GABA releasing cells and made those cells produce these light activated proteins, either one or the other. So after the virus is injected, we then implant a very thin fiber optic cannula in the brains of the mice to deliver laser light to activate the channel rhodopsin or the arch. And that's done because otherwise there is no internal source of light in the brain. So these light sensitive proteins wouldn't work in the darkness of the brain. So we have to insert a thin fiber optic cannula to control the activity of these light sensitive proteins. Okay, so just to recapitulate the whole thing, optogenetics is a method to use light to electrically activate or inhibit brain cells in mammals. And then we can use these proteins that were found in different microorganisms that can use light to electrically activate or inhibit neurons. And then we generate a genetically engineered virus that has the DNA to produce either channel rhodopsin or arch. And we inject that virus inside the brain. The virus will then infect only the cells we want to study because the virus has been engineered to infect on those cells. So it will infect only the GABA releasing cells in the brain region we're studying. Then those cells will then produce either the light activated channel rhodopsin, which in the presence of blue light will excite the cells, or the green light sensitive arch, which in the presence of green light will electrically inhibit the cells. So this is the basic method. So now on to slide 3. So on slide 3, we see how channel rhodopsin works. So we see the inside of the cell and the outside of the cell. So in the presence of blue light, some positive sodium ions go inside the cell, which creates an electrical activation, which are these red little spikes you see at the bottom. So when the sodium goes in the cell becomes electrically active and creates some electrical impulses, which means the cell is more electrically active. And then if those cells become activated, you can see perhaps some behavior changes, right? So if you activate some group of cells in a little nucleus in the brain and then the mouse starts sleeping, then you could say that, oh, higher activity of those cells make the mouse sleep. Then you learn something about how sleep works. So we use this to study eating behaviors. Okay, so now on to slide 4. So slide 4 has a scheme showing how the whole design of this optogenetic experiment works. So first, on step 1, we make a piece of DNA that has the genetic information to produce these light sensitive proteins that were found in microorganisms originally. Then we make a virus on step 2 that has that DNA inside, the DNA to make these light sensitive proteins. Then on step 3, we inject this virus in the brain region we want to study in the rat or a mouse. Then those viruses will infect the cells we want to study and not other cells. Then we implant a little fiber optic cannula that is coupled to an external laser light source, which is this wire you see on step 4. So that fiber optic cable will bring laser light inside the brain region that we were studying. Then on step 5, you see that blue light is coming through that fiber optic cable. The blue light will then activate that blue sensitive channel rhodopsin. That the channel rhodopsin will let in positive charges inside the neuron, and then when the neuron becomes more positive, it becomes electrically more active. So you can see when there's higher electrical activity of those cells, what type of behavioral effect you can see. And then you could say that that behavior was caused by the electrical activation of the cell we wanted to study. So it's pretty neat. It allows us to use laser lights to turn on and off different types of neurons in the brain, and then you can see instantly if the manipulation caused some change in behavior, which could be something like maybe the mouse starts eating more or eating less or starts fighting with another mouse, or maybe it jumps or eat or sleeps. All sorts of different behaviors could happen depending on which neurons you are controlling. Okay, so now on to slide 5. On slide 5, on the upper left, you can see a little photo of what a rodent looks like that's being used for an optogenetic experiment. So you'll see there's a little fiber optic cannula that brings laser connected to a laser light source. Then the laser goes inside the brain, which is shown on the scheme on the right. Then that blue light will activate electrically the cells that were infected by the virus, which is the cell with the red nucleus, was infected by the virus. So that cell will produce channel rhodopsin, and the channel rhodopsin will absorb the blue light and let in positive charges inside the cell, which will electrically activate it. Then on the lower left, you see a little squiggly trace in black. So this is the electrical activity of one particular neuron in this rat brain that has this excitatory channel rhodopsin. Then you see that when the blue light is turned on, which is the little blue line above on panel C, there are more little vertical spikes on that trace, and each vertical spike is a electrical impulse that that neuron released. So the neuron becomes more electrically active, is communicating more strongly with its other neuronal partners, and then you can see if that increased neural activity of that particular type of neuron creates some behavior. And of course, this is not happening with one cell, but at the same time, this is happening with thousands of cells that were infected by many copies of those viruses we injected. So we're not just manipulating one cell, but rather thousands of cells with this laser delivery method. So this is how deep optogenetics works, and this is the main method that's going to be used here. So basically, in the presence of blue light, we can activate the cells we want to study, and then we can see the changes in behavior that this causes. Okay, so now on to slide 6. So that's the kind of method we'll use. So on slide 6, now the proper talk will start, which is that usually on the left, you'll see that a lot of scientific talks have this structure, which is that they'll say that we read a lot of papers and then we're very smart, and then based on these papers we read, we predict some very specific experimental outcomes. And then when we did the research, all the predictions are observed, so we are very good at predicting. But in this talk, the opposite happens, which is that based on prior data, we made a few predictions. Then we saw none of the predictions were observed, so we were very sad. But then after that, we saw some unexpected results, and then we followed up on those and found these new discoveries we'll talk to you about now. So our conclusion are that we are very bad at predicting, but the brain is still interesting and we can learn important things if we follow up on unexpected discoveries. So now we go on to slide 7. Okay, so the region in the brain we're studying is a region in the brainstem part of the midbrain. And this region in the midbrain brainstem is called the periaqueductal gray, which will be abbreviated PAG or PAG in all the other slides. So we're studying this periaqueductal gray region. And the reason why we want to study it is because this region is known to be very important in panic and fear responses. And our lab has studied panic and fear for a long time, so it makes sense that we're interested in studying this region. So, this periaqueductal gray region exists both in mice and in humans in the brainstem. So, in a human, if you put an electrode on the periaqueductal gray of the human and then electrically activate it, the human will develop a full-blown panic attack immediately. So a previously happy person, if you activate their periaqueductal gray with an electrode in a medical setting, they will become very terrified. They'll say they're afraid of dying, that there's something chasing them, their heart rate will go through the roof, they feel like they're suffocating, they really feel like something terrible will happen. So activating this region causes a panic attack in a human. And then in a mouse, activation of this region causes something similar, which is the mouse gets very freaked out and stressed and scared. So on panel D, we are seeing the neural activation pattern of periaqueductal gray cells when a mouse escapes from some dangerous thing. Okay, so each little row, you have 57 rows on panel D, each row corresponds to the electrical activity of one cell in the PAG. So I have 57 cells. And that time course is from safety on the left to when they start escaping from a threat that appeared. So you see this dotted line on panel D. This dotted line is when a threat appeared and then the mice started escaping from the threat. So they are now scared while they're escaping. So you see that when the escape starts, each row becomes generally brighter colored, and a brighter color here means higher electrical activity. So these cells get more active when there is uh, increased escape, which is a panic related behavior because you want to escape from the threat very quickly. So this shows that the cells in this region in general are activated during escape. Then on the panel on the right, these authors used optogenetics and showed that whenever there's higher laser intensity of optogenetic activation, which is on the x-axis, the mouse displays a higher uh, speed of escape. So these cells are both normally activated during escape, and then if you activate them on purpose, it causes escape, which is a rodent measure of panic-like behavior. And these manipulations were done with excitatory cells in this periaqueductal gray region. So basically, if you excite these excitatory cells in the periaqueductal gray, which are cells that, when active, increase the electrical activity of other cells, it causes these panic behaviors like escape and such. So this was known, and then in parallel in humans, if you activate this region without any specificity and activate all the cells in the region, it causes panic in a human. That also was known. So here, this data on slide 7 is done with excitatory cells of the periaqueductal gray. So we thought that, well, if the excitatory cells in this region cause panic, then probably if we activate the inhibitory cells in this region, that might decrease panic or inhibit panic, decrease fear, something of that sort. So that was our hypothesis, basically that the inhibitory GABAergic cells, which release the neurotransmitter GABA, will suppress these threat-induced actions like escape and panic. Uh, so just one term here, which is that GABA is the main inhibitory neurotransmitter. So that's why we are studying these GABAergic cells, because the excitatory cells cause panic. So we thought the inhibitory GABAergic cells would suppress panic. And VGAT is the name of a particular gene that is expressed in these inhibitory cells. So the cells we are studying will be called periaqueductal gray GABAergic VGAT cells. So we thought these cells would inhibit panic behaviors. So we predicted that these cells would, inhibit panic behaviors and that they would represent the proximity of the threat. So those are our predictions. And then on slide 9, we tested those predictions. So we use optogenetics, which is the method we mentioned before, and we excited or inhibited these GABAergic cells in the brainstem while exposing the mouse to two different types of dangerous environments. So on panel G, you can see a little figure of a maze in the shape of a cross sign. And then in the maze, you can see that there's two closed locations and two open locations without walls around them. So the open locations are kind of like just planks suspended in the air. So mice are prey animals, so they try to hide from dangerous things. So generally, they'll prefer staying in the closed, dark areas of this maze in G and they'll try to avoid being in the open space. And if you have seen a mouse in real life in the wild, you might have noticed that the mouse is generally hiding under some dark location in a corner, and it's rare that the mouse is just running around in the open space that's brightly lit, because they're more susceptible to predators in these open spaces. So the mouse will spend more time in the closed spaces, and the amount of time he avoids the open spaces is a measure of anxiety in mice. So we see that if we optogenetically excite these VGAT GABAergic cells in the midbrain and brainstem with optogenetics, that doesn't change their how scared they were of the open spaces. And if we inhibit these cells, it also doesn't change how scared they were of the open spaces. So that was a negative result, and we're pretty sad. Then on panel J, we exposed the mouse to another different type of threat, which is a live predator. And the live predator is a rat that you can see on the right side of the panel J. So the mouse is a prey to the rat in the wild. So the mouse is afraid of the rat, which is much bigger than the mouse. And then we measured many different things, the mouse behavior when we exposed them to this threat, such as how quickly it escapes, how much it avoids the rat, etc. But neither optogenetic activation or inhibition of these cells change any behavior that the mouse displayed when faced with this live dangerous predator. So these cells appear not to uh, change any threat induced panic, fear, escape type of activity. So, which is our prediction. We thought these cells would be inhibiting these panic behaviors, but it seems like nothing happened. Okay, then on slide 10, we tried to see if these cells were more or less active when the mouse was more near dangerous things. So again, on the left side, you can see the two threat assays we use, which are the open spaces on the maze on the top and displaying the behaviors while in front of a live predator, which is the rat on the bottom. So on panel M, we plotted the neural activity of these cells we're studying in the midbrain, and warmer colors represent kind of higher neural activity. So the rat is on the right side where it's indicated. Then we don't see warmer red colors more near the rat. Neither do we see more red colors in the dangerous open arms of the maze compared to the closed arms. So these cells don't seem to particularly care about where the threat is. They're not more or less active when they're near the threat. So the activity of these cells doesn't seem to represent distance to the threat. And when we inhibit or activate these cells, no fear, panic related action was altered. So basically, our predictions completely failed, and we didn't see any fear related behavior being modulated by these cells. So then we were very sad and thought we should discard the project and do something else. But we noticed that when you activate these cells, the mouse gets a little more exploratory. So it starts maybe nibbling on little things and trying to look around what's happening more. He seems more exploratory when we activate these cells. So we thought that perhaps these cells might be involved in food seeking. And now we go on to slide 11. Then on slide 11, basically we looked and read more about this food seeking related circuits, and then we found that this region we're studying, which is the periaqueductal gray, it receives electrical inputs from many different regions in the brain, such as the amygdala and stria terminalis and lateral hypothalamus. And activation of many of these different inputs to the periaqueductal gray region that we're studying caused increased food seeking behaviors. So we thought, oh, so it is known that some inputs to this region can cause food-seeking behavior. So perhaps the cells we are studying, they might cause food-seeking behavior. But no one has ever found any cells inside the periaqueductal gray region or PAG region that causes food seeking. So then that's what we tested. So first we try to record the neural activity of the cells we're studying, which you can see in slide 12. So slide 12, you see the two assays we used on the left side, which is we expose the mouse to either a walnut or a cricket. So the walnut is a very fatty, caloric food. Walnuts are very fatty. And the cricket is also very caloric because it's very high in protein and fat. So cricket and walnut are very tasty foods that the mice likes a lot. And then we record the neural activity of these cells in the brainstem. So on panel A, you see a lot of little traces in pink and purple. So each little trace is the neural activity of one particular cell in this nucleus we're studying. And it's the neural activity of many different VGAT GABA releasing neurons in this region. So you see some 20 or so traces, which are the neural activities of those cells. And peaks in these traces represent moments in time that those cells were more active. So you can see that different cells are active at different times, and the recording of neural activity is working. So that's on slide 12. Now we go on to slide 13. And on slide 13, we panel B, you can see some other traces of neural activity of, and then we see what happens when the mouse is eating the cricket or the walnut, and the eating epochs are shown in blue on panel B. And basically what we observed is there is higher neural activity, meaning there's more peaks before eating. And then when the eating starts in blue, you see that in general the traces tend to be more flat, like there's fewer peaks during the blue times than the non-blue times. So the non-blue times are when he's foraging and looking for food, and the blue times is when the eating happens. So basically these cells seem to be more active during the approach to food, and then the activity decreases once he starts eating the food. Okay, so on panel C, you can see an example of that, which is this purple trace on C is the neural activity of a particular cell. And then at the red dotted line, that's when eating starts. So you see that the cell is more active prior to eating, and then when eating starts, the activity drops. So these cells are more active during the food seeking moments, and then when eating starts, the activity drops. So because the cells are more active during the food seeking moments, we thought that, well, perhaps these cells have something to do with causing food seeking. And now we go to the slide 14. So slide 14, what we did is we had recorded the neural activity of many of these different cells while the mouse was either eating crickets or eating walnuts. And then we observed that there are different types of neural activity when the mouse was eating those different food sources. And we could, just by looking at the neural activity, predict if the mouse was either approaching the food sources or eating the food sources. So that means that we could just look at the neural activity, not look at the behavior, and then from the neural activity, we could estimate pretty well what the mouse was doing. So, and then if you go back to the video and watch, we can see the prediction usually correct. So just with the neural activity, you could say if the mouse was eating or approaching, because the neurons have kind of different types of neural activity during those different behaviors. So basically, this data showed that these neurons are more active during the food seeking time points, and then their activity decreases once they find the food and start eating. Okay, so now we go to slide 15. Then on slide 15, there are two links for YouTube videos. So just first click on the first upper link in blue on the right, and please open that video. So I'll just wait a little until everyone opens that video. And now play the video. So what you'll see is a optogenetic experiment where a mouse is walking around, and when there's a flashing blue light on top of the mouse's head, he starts eating the chocolate very quickly. So one thing you notice here is that on the left there's a food pellet that is not very tasty and is not chocolate, and then on the right there's the chocolate. So when the blue light starts flashing, which is when we're optogenetically activating those cells, he immediately goes to the chocolate and starts eating it. So that showed that the activation of these cells caused eating. Okay, so now please click on the second link on the right on slide 15, and then please open that video. And on this video you can see that the mouse is chasing a little cricket, and during the off times when we're not stimulating this brain region, now the mouse has some interest in the cricket but is not super motivated to eat it. But then as soon as the laser gets turned on, which you can see with the blue light, then the mouse immediately goes next to the cricket, tries to hunt it, and gets the cricket and eats it very quickly. So in both cases, you can see that the optogenetic activation really increased the approach to the food and then the subsequent consumption. And that's the behavior that is plotted on G and H. So we're showing that the optogenetic activation of these cells decreased the amount of time it took for the mouse to eat the cricket, which is the first graph on G. And then they ate more walnut when we activated these cells with channelrhodopsin, which is the red bars on all the graphs are the channelrhodopsin graph bars, and the gray bars are the control mice in which we did not activate these cells. Uh, so the activation of these cells decreased the amount of time to get to the food, so they got to the food more quickly, and activating these cells increased the amount of cricket eaten and walnut eaten. And one important thing is that here the mice were not hungry. So these are mice that just ate a big meal, so they're definitely not hungry. But if we activate these cells, even in these non-hungry mice, you could see it causes voracious eating and approach to these caloric food sources that are high in fat and high in protein. So that's one important result we found, that activation of these cells causes voracious eating even in non-hungry mice. Then on 16, slide 16, we started further characterizing what the activation of these cells do. And to do that, we put the mouse in this kind of square board that has little circular holes in it. And mice like to put their face inside the holes to see what's happening, and they like very exploratory. So this is a maze to measure exploratory behavior. And there's no food here, but they can explore and try to see what's inside each hole. And there's nothing inside each hole, but they like to explore and see what's inside the hole. And there's like 16 little holes in there. And what we see is that when we optogenetically activate these GABAergic cells in the periaqueductal gray region, the mouse does more head dips inside the holes to explore and see what's happening. So this manipulation both causes higher food eating, as we see in slide 15, and more exploratory behavior. Then we go now to slide 17. So here we made this experiment shown on the upper left. So it's a square box, and then on opposite corners of the box we hung by a string either a wooden object that cannot be eaten or a walnut which can be eaten. So it's a fourth source of food. So there's a food and a non-food object hung in the ceiling of opposite corners of this box, and they're high enough that the mouse is not able to eat the walnut. So it's as if you're home and there's a chocolate bar pasted to the ceiling, and then you can see it, but you are not able to reach the chocolate, but you can see it and smell it. So there's this walnut that is hanging above the mouse, but it's too high for the mouse to reach it. So we wanted to see if the activation of these cells, and these are also in not hungry mice, activation of these cells would cause kind of approach to the walnut in a way that's separated from the feeding. And that's what we found, that if we activate these cells, the mouse spends more time near the underneath the walnut than it does underneath the object. So the activation of these cells increases the interest to be near the walnut, even though they can't eat the walnut and they're not hungry. So even though they're not hungry, activating these cells causes this craving to be near this food, even though they cannot eat the food because the food is kind of out of their reach. So that's what's shown on slide 17. Then on slide 18, we try to see if we activate these cells, will the mouse do some kind of more complicated action to gain access to the food? So in the previous slide, the walnut was hanging, but it was outside the reach of the mouse. And now the walnut is also hanging, but the mouse can reach it if it uses this little ladder to climb and eat the walnut. So the walnut is also high, but now the mouse can climb to eat the walnut. And we see that activation of this cell decreased the amount of time that the mouse takes to climb and eat the walnut, and it increased the amount of walnut eaten. So this shows that um, the activation of these cells can cause some slightly more complicated actions to get the food. So the food here is not accessible readily. The mouse has to figure out that I have to climb this ladder to gain access to the food. And then activation of these cells causes enough craving of the food that the mouse is able to figure out how to do this to get the walnut, even though they're not hungry. Okay, so this suggested that um, perhaps the mouse might be in a state of higher positive motivation when we activate these cells. And then we also observed on slide 15, like on 15H, you can see that activation of these cells increased hunting of crickets. And the crickets are living insects, and the cricket does not want to be eaten by the mouse. So the cricket wants to run away from the mouse. So the mouse has to run after the cricket on slide 15 to eat the cricket. So one part of hunting the cricket is to follow or pursue or chase the cricket. So chasing is a type of behavior they have to do to hunt the cricket. You have to run after, chase the cricket. So we wanted to see if activation of these cells caused enough interest in chasing that maybe it could even make the mouse chase some object that's not even a source of food. So on slide 19, there is a little YouTube link there. So please open that YouTube link on slide 19. And then on that link, we see that what we did basically is we put a little plastic ball on a stick. And when we optogenetically activate these cells, which you can see on the right image, the mouse follows that ball as if it's trying to hunt it, even though it's not a source of food. And the control mouse on the left does not follow the ball because it's not hungry and has no interest in ball. So this shows that the activation of these cells causes uh, chasing, which is a type of behavior the mouse has to do to chase and hunt the cricket. And it does this chasing even towards things that cannot be eaten. Perhaps it's inducing the craving towards finding and chasing some caloric food source. Then on slide 20, you kind of show the quantification of that effect, and basically showing that the optogenetic activation of these cells increases the proximity and following towards this plastic ball that we're moving around. So that's slide 20. Then on slide 21, we wanted to see if the mouse kind of liked or didn't like activation of these cells, right? So you could imagine that activation of some circuit in the brain might be very unpleasant because maybe it causes like pain or hunger or some other unpleasant state. And you could also imagine that there's some other brain circuits that when you activate cause some good, positive, reinforcing, pleasurable feeling in the brain. So there are brain circuits of both types, and both types of feelings can induce increased eating. So one reason that a brain circuit might cause more eating is because it's causing increased hunger. So increased hunger will cause more eating, but hunger is very unpleasant, right? And another reason to cause increased eating might be some positive craving of wanting to get caloric, tasty rewards, which could be a good feeling. And we don't really know what the mouse is feeling, but we can devise a test to see if the mouse kind of likes to have this stimulation of these cells or doesn't. And that's what we try to measure here. So on the image, you see there are, so just look at the first upper left image, which is this rectangle here. So the rectangle is a box that's divided into two halves, the left half and the right half. And then the mouse can freely go around to the left or the right side of the box, and there's nothing particularly interesting in either side. And the mouse can explore and go to one place or the other. And in the control condition, which is the first uh, rectangle, the upper rectangle, you see the mouse spends equal amounts of time on the left and the right. And the amount of time he spent on each pixel is plotted in these colors. So red means more time spent in that pixel, and blue means less time spent in that pixel. So roughly you see there's equal amounts of blue and red on the left and right side of the upper rectangle, which is a control condition. So the control mouse, where you're not stimulating these cells, then the mouse doesn't particularly prefer being the left or the right side. He spends equal amount of times in the left or right side of the box. Then the lower rectangle, we optogenetically activated the cells only when the mouse entered the right side of the box. And then you see that there's much more red and yellow color on the right side of the box in the lower uh, image, which means that whenever the mouse went to the right side of the box, there was optogenetic stimulation of these cells. And if that stimulation was good or the mouse liked it, it was something pleasurable, then the mouse would spend more time in the right side of the box because that's where the stimulation is happening. And and that's what we see, which suggests that this stimulation is something good, liked, pleasurable, positive, etc. And this also shows that the stimulation is not something that's causing a psychologically negative effect in the mouse. Because if this optogenetic stimulation caused very intense hunger or thirst, which is very unpleasant, then the mouse will try to avoid the stimulation. But because the mouse spends more time in the location that they get the stimulation, it shows that whatever the stimulation is doing to the psychological state of the mouse is something likely positive that they like or want, and it's not something unpleasant like thirst or hunger. So that's slide 21, which shows that the activation of these cells is almost certainly not causing hunger, but instead is maybe causing something that is positive, such as the anticipation or craving of getting some tasty food in the future, something like that, perhaps. Okay, so now slide 22. So here in slide 22, we try to study if the excitation optogenetically of these cells could induce compulsive eating. And compulsive eating is something that's seen in many eating disorders. And one common way to define compulsive eating is to eat despite the presence of negative consequences. So if you overeat, you can have some negative consequences in your life, and then if you continue eating while having these negative consequences, that is classified as compulsive eating. So you keep doing the behavior compulsively even though negative things are happening. And then one way we can study that in the mouse is in this assay here. So on the left, you see the drawing of the behavior, which is there's a box, then the mouse is placed on the left side of the box, and then on the right side of the box, there is you see the stripy floor, which is a metal grid. And then on top of the metal grid, there is a walnut. Okay, so the mouse has interest in eating the walnut, so the mouse goes on top of the metal grid, which is on the right, the stripy parts. and then eats the walnut. Okay, so on the graph, you can see the amount of walnut consumed during baseline, which is when the mouse is not being shocked, nothing's happened, so the mouse can go onto the metal grid and eat walnut. So you see that the gray control mouse and the CHR2 expressing optogenetic excited mouse eat the same amount. And in this condition, baseline, we're not activating the cells, but both groups of mice like to go on top of the grid and eat the walnut. Then on the shock period, we then give a very weak electric shock whenever the mouse goes onto the metal grid. So now the mouse still wants to eat the walnut, but whenever it goes to the near the walnut, it gets a little weak electric shock. So the mouse doesn't like the shock, so it decreases the amount of walnut it eats. So you see that the amount of walnut eaten during the shock epoch, both the gray and the pink bars decrease in size compared to the baseline, meaning that in the absence of shock in the baseline, they eat more walnut, and then if the walnut eating is associated with shock that are electric, then they don't like it, then they eat less, which makes sense, right? So imagine that whenever you went to the kitchen to eat a chocolate, you got a little electric shock, then you'll eat less chocolate. So this makes sense. This is a negative consequence of the eating. Here is this electric shock. Then on the third part of the assay, the electric shock was still happening, but we also use the laser light to optogenetically activate the cells. Then you see that the red bar in the shock plus laser epoch increased compared to the shock epoch, which means that if we activate these cells, they're willing to endure the electric shocks to eat the walnut. So their electrical activation of these neurons with optogenetics causes eating despite the negative consequence of having electric shocks being given to them. And this result basically shows that the activation of these GABAergic neurons in the periaqueductal gray region induce compulsive eating, which is they induce increased eating even in the presence of something aversive like electric shocks. So that's this result on slide 22. Okay, now let's go to 23. So 23, try to see in more detail what types of food are eaten when we activate these cells. And each graph here is one particular food, and here we're plotting only highly caloric, sugary, fatty foods. So the first graph is chocolate. Then compare the gray bar, which is the control. You see that activation of these uh, GABAergic cells in the brainstem periaqueductal gray region causes a increase in eating chocolate. It also causes increase in eating cheese, which is the second graph, increased eating of sausage, the third graph, and increased eating of sugar. So activation of these cells seems to increase the eating of chocolate, cheese, sausage, and sugar, which are all very highly caloric, kind of not super healthy foods. And this happened in non-hungry mice. So these mice are not hungry. They had just had eaten a lot of their normal food chow. And even though they're not hungry, activating these cells greatly increase the amount of these very highly caloric, unhealthy things that we gave them, compared to the control mice in which we didn't activate these cells optogenetically, which is in gray. So basically, the red bar shows that when you optogenetically activate these cells, they eat much more of these foods compared to the gray bar, which is mice that did not receive this optogenetic activation. Okay, so activation of these cells causes more eating of these fatty caloric foods, but does it cause more eating of healthy, nice foods as well? So we did that in slide 24. And then on slide 24, we see that activation of these cells did not increase the eating of vegetables and fruits in non-hungry mice. So these are also non-hungry mice, just like in slide 23. And non-hungry mice will eat more of those chocolate cheese things if we activate these cells, but they don't eat more of vegetables like carrot or fruits like cherry when we activate these cells. So these cells, when activated, seem to cause the increased consumption of cheese, chocolate, and sugar and sausage and everything that's caloric and fatty and sugary and bad, but it doesn't seem to increase the eating of carrot or cherry, which are maybe healthier vegetable alternatives. Okay, so now we go to slide 25. So remember from the introduction, I had mentioned that this optogenetic activation can be done with uh, channel rhodopsin. And we can also do optogenetic inhibition of cells using this other tool called ARCH. So ARCH, in the presence of green light, will cause electrical inhibition of these GABAergic cells in the periaqueductal gray region. And these are hungry mice. So now the mice are hungry, so they haven't eaten for 12 hours, and so they're very hungry, and they should be wanting to eat because they should be hungry. And they eat some amount, but we see that when we inhibit these cells, they eat less than the control mice. So they take longer to approach the food and they eat less, showing that the food seeking behavior is decreased even in hungry mice when we inhibit these cells. So that's the last result we had. And then the conclusion is on slide 26. So the first conclusion is that we observe that these GABA releasing VGAT cells in the periaqueductal gray region, they are more active during the approach to food, and then their activity gets inhibited during the eating, consumption, swallowing. And we show that if we activate these cells, they eat more food even if they're not hungry. If we inhibit these cells, they eat less food even if they are hungry. And the activation of these cells also causes more exploration, and it seems to be positively motivating because the mouse likes this activation. And it also causes more compulsive eating, which is eating despite the negative consequences of electric shocks. And lastly, we showed that the optogenetic activation of these cells causes more eating of sugary and fatty foods like sausage and sugar and chocolate, but it doesn't cause more eating of fruits and vegetables. And these experiments were done with non-hungry mice. So even if we get a not hungry mouse that's fully satiated, activating these cells can still induce the eating of these very highly caloric things like sausage and chocolate, etc. Okay, so now the final conclusion is slide 27. So in 27, we showed that basically, if you get a not hungry mouse and activate these cells, they eat more of these caloric foods. So they can cause excessive food seeking, which is something you can see in binge eating disorder. And then we also see that when the mouse is very hungry, he is food deprived, so he is very hungry, and then we inhibit these cells, then the mouse eats less, even though he's hungry, which is like an anorexia like symptom, eating less even though we're metabolically in need of food. So the overactivation of the circuit could be involved in the production of symptoms of binge eating and obesity, while the underactivation of these cells might be something that's happening in patients that have anorexia. But these, of course, are done only in mice and not in humans with anorexia. So these views have to be tested in future studies, but that is what our studies suggest might be happening. And then on slide 28, this is a picture of our lab members that are all very happy in the sunshiny weather of California. So this work was done predominantly by Fernando Reyes, which is this scientist here with the black beard. And then on slide 29, we'd like to acknowledge the institutions that gave us financial funding support to make these experiments. And on slide 30, I have the opposite, which is a negative anti-acknowledgement slide, where these are foundations that didn't want to fund us. So, boo, bad foundation didn't want to fund us. Slide 30. And then slide 31. Now, I'm happy to take any questions about this result, and these are the conclusions as written again. And if you wish to follow our work further, you can follow me on Twitter on this link, or you can write me an email on this email address on slide 31. And our lab website is the last link on slide 31. So now I'm happy to take any questions from the audience. And thank you again, Cecile, for inviting me to share this work with everyone.
Thank you, Avi, for the very detailed presentation. I think you are the first speaker who had here that whom we had invited in quantum photonics who had explained optogenetics in great detail. So thank you, and I hope those who weren't able to catch it would listen to the replay on the beginning of the talk of Avi. So yeah, we'd like to announce to the audience that. Avi is ready to answer any questions. Please raise your hand. We will bring you up the stage if you have some questions or clarifications, and Avi will take these questions. Yeah, so yeah, we are waiting. So anyway, I'd like to just comment, Avi, you are the
Mhm.
first a group whom I had seen presenting a study with the negative acknowledgments. So you are giving me an idea, and I'm quite amused about it because, yeah, I think the study that you have is very important and that it should be given, funding and support. So anyway, the first question, since
Mhm.
I'd like to ask you, is how do you think is your finding related to other behaviors, like, for example, addiction or obsession? Do you think this part of the brain, which either when understimulated or stimulated, is it related to such behaviors? Because here, basically, I know that you serendipitously tried to discover the eating behavior because you were studying pain and fear. Am I
Mhm.
right? Can you please comment on this?
Yeah, that's a good question because these data, we didn't study addiction here, but our data suggests that these cells control some behaviors that are abnormal or seen in addiction, like compulsive behaviors, right? So people that are addicted to drugs will do that action despite negative consequences. So there's compulsive drug seeking. And this is something we're interested in exploring in the future, which is to see if these cells have some role in drug seeking behaviors as well. But we haven't done that yet. But maybe they do. And then I'll tell you in the future.
Yeah, thank you. So I'll give the mic. Hi, Kevin. Welcome to the stage. I will give the mic to you. You are a priority. So, yeah, Kevin.
Oh, hi. Well, I might have been sort of hoping I could Google it first, but and cheat. But yeah, maybe I'll just ask. In all seriousness, I don't know if they have discovered, genes for curiosity. You know, because basically I'm thinking, intellectual. I'm serious. Intellectual curiosity. Because it's well known. But that the health problems, so I mean, even with, just to really know how what you're doing to your body, so all I can think of is, yeah, I don't know if they can highlight the genes for intellectual curiosity for people to for some people to override their impulses and such like that, I guess.
Uh, well, here we didn't study anything about genes related to curiosity per se, but what was related to that is, so I didn't mention these data, but we also found some other results showing that these cells we're studying, they are very strongly interconnected with this other brain region called the zona incerta, and this other brain region has been shown to be very important for exploratory curiosity in mice. So activation of this region that is very connected to these cells we are studying, that other region eh, is known to induce this exploratory curiosity. So this circuit could be related to that through this interconnection.
Oh, interesting. Oh, sorry, audio problems. Yeah, okay. I can't wait to look that up. Thanks a lot.
Yeah,
Wow.
thank you.
Yeah, thank you, Kevin, and thank you, Avi. So, Bob, is it okay if I request you to read the question here in the room chat?
Sure thing. This is from R, and he first says, "Great talk, doctor." He has a couple of questions. Did you monitor the natural habits of mice in a non controlled environment with the same walnut or chocolate placed instead of in the glass box?
Sorry, did I do what?
Instead of in the glass box.
But sorry, so you said the whole question again. Did I do what?
Oh, okay. Um, did you monitor the natural eating habits of mice in a non controlled environment with the same walnut or chocolate placed instead of in the glass box?
Yeah. So the normal behavior of the mouse is to want to eat some amount of chocolate or some amount of walnut, even if not hungry. So, but they eat much more chocolate and walnut when not hungry if we also activate these cells while that is happening. So, if you just put some chocolate in its home cage while not hungry, which is its normal environment, then it will eat some amount of chocolate while not hungry, just like we would eat some chocolate when not hungry. But yeah, activating these cells dramatically increases this amount of extra calorie they're eating when not hungry.
I thank you. Now, question number two. Did you repeat this experiment in total darkness?
We did not, but I think it would work because the mice have very good sense of smell. So a primary way that they are um, finding the food in the environment is through the sense of smell anyway. So probably it would work in the dark, but it might take a little longer. So the walnut will almost certainly work in the dark, but the cricket might not work in the dark because the cricket is running around and trying to escape, and then they do use their vision to hunt the cricket. But the walnut or the chocolate or the sausage or these things are not moving and they have very strong smells like the sausage. They almost certainly this would work in the dark, but perhaps not the cricket.
So I thank you. Now, R has two more questions, but I think I'll pass the mic to Mustafa. Good evening, sir. And we'll circle back in just a second.
Okay.
So are you with us, Mustafa? Yes.
Yeah, thank you, Bob. Thank you, Cecile. Thank you very much, Dr. Ivy, for your great talk. Uh, I have many questions, but
Mm-hmm.
I apologize for my bad English. Uh, to be honest with you, I write down my question in French and use Google Translate. So, first question.
Okay.
First of all, the application of optogenetics are impressive, particularly in terms of understanding the brain and in the treatment of neurological disorder.
Mm-hmm.
However, the safe and effective introduction of genes coding for opsins into neurons remain a challenge,
Sorry,
especially
say
for
again. What remains a challenge?
uh, introduction of genes coding of opsin
Mm-hmm. Yes.
into neurons.
Okay.
It remains a challenge, especially for clinical application in human.
Mm-hmm.
What do you think are the way or approaches to overcome this challenge?
Okay. So first, I'll just explain your question to the listener. So he mentioned that here we used optogenetics to study these circuits, and then you can find lots of new discoveries in mice and other animals using this method. But at this moment, it's very difficult to use this method in humans. And then the question was, how would this be used directly in humans? And uh, at this moment, there, like you said, there is no way to. clinically approved way to do this in humans, and part of the reason is because um, to use this method at this moment you have to inject some kind of virus inside the brain of the mouse, and then the virus will infect the brain cells and make them produce the light sensitive proteins that are used for optogenetics. But that is not really safe to do in humans at this moment, like you said, because we don't know what the long-term consequences are of injecting viruses in human brains of this sort. Because for the mouse, there isn't any uh, safety problem within the lifetime of the mouse, which is only 2 years. But humans live 30 years, so who knows what will happen 15 years later after we inject this virus in the brain. And so at this moment, there's not some safe, known way to use this in humans. And then there's also the problem that even if we have some uh, safe virus that we know works in humans, which you don't, but even if we did, the problem is that you also would have to implant this fiber optic little cable inside the brain, which would be a very invasive thing to do to a patient. People don't want fiber optic cables inside their brain. Um, so even if the virus problem was solved, this other problem is also very big. So it doesn't seem likely that this will be used in some large scale in uh, human patients. But the value is that by using this, we can discover a lot about how different cell types in the brain control different behaviors. So let's say if we find that in this case, oh, these particular cells in this region of the brain control food seeking. And that's likely also true in humans. But then, if we just find some kind of other manipulation that alters the activity of those cells in mice or humans, then that would be the way to target this knowledge to humans. So let's say we find that, oh, if the mouse takes this drug orally, then that drug inhibits the activity of these cells we're studying, leading to less food seeking. Then you could test the human if the human orally taking that drug in a pill might also decrease the activity of those same cells in the human, leading to less food seeking. So that would be one way to apply that knowledge gained from optogenetics without directly using optogenetics in people. And there's also other things. So for it could be that we find in the future that some type of life intervention might inhibit or activate these cells in the mouse. So perhaps we find that if the mouse does a lot of exercise vigorously for 2 months, then these cells get less active, leading to less food seeking. Then we could test if in humans doing, say, increased exercise or whatever is that manipulation also causes the same effect in the behavior we're interested in. So basically, it's hard to see how optogenetics could be used widely as some therapeutical tool in human patients, but it's easier to imagine how the knowledge that we gain by using optogenetics in lab animals can be kind of used to develop therapies that will be helpful for real patients that are people.
Thank you, Doctor. If we exclude a potential application in humans, how are the ethical challenges associated with the use of optogenetics genetics in research on animals models?
Right. So you asked what is the ethical concern of using optogenetics in animals in the lab, not in humans.
Yeah, yes,
Okay. Uh, yeah, so that too is a very important question that, if you think about it, is even broader, which is not specific to optogenetics, but what are the ethical considerations of using uh, lab animals for discoveries in general, using other methods as well. And that is definitely something that we are very concerned about in both here in our university and every other major university in America. So, for example, you are not allowed to just do an experiment in mice. You have to propose what you're doing very carefully and write many steps of what's being done to decrease the pain and discomfort of the animal, and what analgesics you're using, and how the animal is recovering from the surgery, and those sorts of things. And then that protocol has to be reviewed by a bioethics committee that includes uh, physicians and veterinarians and scientists, and also members of the general public. So they all review those protocols, and then they say, oh, here, after the surgery you should also use this other type of painkiller, and then those comments are included in the actually done experiment when it happens. So the experiments have to be very carefully proposed in a manner to decrease the pain, discomfort, etc., to the lab animals. And but you still could argue that it's still a problem, because it does create these discomfort for the animal, even though we are using the painkillers, which is true. But then you have to weigh that against the cost of essentially not developing any new biomedical technologies that will help humans eventually. So those are the kind of considerations that have to be thought about.
Thank you. Bob, can I ask another question, or you prefer?
Yeah, sure. Mhm.
Now go
Okay,
for it.
thank you. Dr. Evie, does your research rely exclusively on optogenetics, or do you combine optogenetics with other imaging techniques to extend the study of the functional and individual
Mhm.
neurons?
So
Uh,
we we use three main methods. So here, optogenetics, the main one that was talked about here. But in the beginning of the talk, we used another method to record the neural activity, which is on slide 12 and 13, where we recorded the electrical activity of the cells just to see when they are more or less active. And that's a method called calcium imaging of neural activity recording. So there's another method. And then we also used this third method that wasn't mentioned here, called chemogenetics. And that is a method to chemically alter the activity of target cells of interest in the brain, and then see how the behavior changes. And this chemogenetics method is something that is easier to imagine how it could be eventually used in human patients, because it doesn't involve some laser light source and a wire and a fiber optic implant. It just involves viral delivery of a gene, which hasn't been done in humans, but I believe that will be solved eventually, because there's a lot of interest in doing that. And but it's not invasive, because the human will just have to take some drug orally to have the chemogenetic tool working in them. So that method would be easier to imagine being applied to real human patients.
okay. Uh, Thank you. Mike to you, Bob. And if there are times remain, I can ask more questions. Thank you very much.
Thank you, Mustafa. I think you had great questions. So I do want to cycle back to R. He has two more questions. The first being, do you have a hypothesis as to why they go for high calorie food only?
Yes. So I believe it's because of the mechanism involved in what these cells are actually doing. Okay, so let's consider some possibilities. So one possibility is that if you activate those cells, it causes a lot of hunger. But then if it causes hunger, it could cause hunger to eat things in general, right? So the fact that the activation causes eating only of some tasty things suggests that it's not inducing more eating due to increased hunger. So it's probably not that. And another alternative would be that it's inducing something like the craving for tasty, rewarding foods. And then these, chocolate is tastier, more caloric, more rewarding. So that seems to be better supported by our data. And then we did a new experiment that I didn't talk about yet, but is ongoing in the lab. So here we got some hungry mice. So the mouse is hungry, wants to eat food. And then we put them in a box where there is two foods. There is on one side a accessible kind of mouse food chow, which is their normal food that they eat every day, but it's not some tasty, interesting food. But that's a food that's there, accessible, which the hungry mouse should want to eat because it's hungry. And on the other side of the box, there is chocolate, but the chocolate, they like chocolate more than their normal chow. But the chocolate is inaccessible. Like the chocolate is inside a little cage. So they can see and smell the chocolate, but they can't eat it. So then if you put a normal hungry mouse in there, it goes near the chocolate and sees I can't eat the chocolate and just eats the accessible, boring chow food because that's something to eat and he's hungry. But then something interesting happens when you activate these cells and put them in that box. Then the mouse is hungry, but instead of eating the accessible, boring chow food that's on one corner, the mouse just stays near the non accessible chocolate, which suggests that the craving for this uh, or anticipation of getting this tasty food is what's driving the behavior. Because they will, while hungry, ignore the real food that they could eat and prefer to spend time near the caged chocolate that cannot be accessed. So we think the reason that the activation causes the eating of these tasty foods only is because it's increasing some kind of either craving for tasty foods or some anticipation of the pleasure of getting a tasty reward, something like that.
All right. So,
But those
our
are not
yeah.
really well uh, resolved issues, so we're still kind of doing new experiments to see what's happening.
Ours fourth question I find particularly interesting. He states that GABA is a stress reducing agent, and he asks if does a reduction of stress induce peculiar eating habits?
Does reduction of stress do what?
Does the reduction of stress induce peculiar eating habits?
Well, we see more the opposite, which is the increase of stress can change eating habits, and then the decrease might go back to where it was before. So uh, it is also a little complicated because the increase in stress in different people can cause either more or less eating. So there are people that eat less while more stressed, and there are people that also eat more as a measure of comforting during more stress. So both things can kind of happen. So it's not something that is super well studied in these kind of mouse models. In the mouse models, what's more studied is eating during acute danger, which would be something like there's a mouse, then there is some live predator, which is in real concrete danger at that moment. And then generally the mouse will eat less because it wants to minimize the exposure to this danger. Um, but the more interesting question that you're probably getting at is what happens under chronic stress, which is something chronicle is always badly happening, like your job is stressful or you have economic insecurity, and in that condition what happens to the food seeking. And that is, in humans both things can happen. You can eat more or less in different people in that condition, but in mouse models that really hasn't been studied well.
All right, I want to welcome LT. How are you this evening?
Thank you, Bob. It's beautiful presentation. Um, I have a question.
Okay.
Avi, yes,
Yes.
you can hear me all right,
Yes.
right?
Hmm. Hmm.
So the mouse, I understand that there are some like diabetes mouse models. Would you use those mouse? Have you tried with those mouse with this method?
We have not, you know, yeah.
Mm-hmm.
Essentially, everything we did is we presented now. But this is just some new things that we just concluded this study, 1 month ago. So now this opens a lot of new directions, such as some of which you people already insightfully suggested, such as looking at what will happen during addiction behaviors or in mice that have some metabolic problem, like mice with diabetes models, uh, or how it affects this curiosity. So there's a lot of things that you already suggested here that would be interesting to look into further.
Okay, thank you. Look forward to your next presentation then. Thank you.
Yes, thank you.
All right. So Ayman has a question. What if there's no food around, but you stimulate the mice? Have you experimented with that situation?
Hmm. Yes, that we have done. So if there's no food around and stimulate the mice, they become more exploratory. So they stand up more to see what's happening in the upper parts of the wall. They sniff more things. They go into other places. They become much more. Let Let's see what's happening. They look more curious and exploratory in the absence of food, and if you stimulate these cells. So they do that, and we also saw that this stimulation in non-food conditions, when there's something not there isn't any food present, it doesn't cause more social interaction. So they're not more curious about another mouse, but they do get more curious about the environment. But they neither attack nor interact more with another mouse if we activate the cells. So it seems not to be related to social aspects.
And a follow up question from Eamon. Does the amount of food relate in any way to the level of their movement activities?
Does the amount of food? Just explain that again.
Does the amount of food relate to the level of their activities, movement activities? Do you feed them more? Are they more active? Feed them less active.
Oh, I get it. Yeah, the so it depends on how much of that happens. So basically, um, if they are very satiated and not hungry, they'll be generally more lethargic, less active than a slightly hungry mouse. Just like we would be, after eating a very big lunch, you are more lethargic, less jumpy, and then the mouse is slightly hungry. It will be more exploratory to try to find more foods and etc. But then if the mouse is extremely hungry, not a little hungry, but extremely hungry, it will then go into kind of energy conservation mode. So then it will retract and move very little and drop its body temperature and only do movements if it's reasonably sure that they'll get food by doing it.
I should ask if Kevin or Mustafa or LT have additional things that you want to get back to.
Bob, before anything else, there was a question which was sent to me
Mm-hmm.
by Verna, and I'd like to read it. So, the ability of mice with activated VGAT PAG cells to endure food shocks or to endure food shocks to obtain fatty foods suggests a powerful motivational drive. how might this circuit interact with other brain regions involved in reward processing and decision making? And what implications could this have for understanding the mechanisms underlying impulsivity and addiction?
Hmm. So these are all again things that we didn't do, but our current hypothesis would be that these cells would be somehow reinforcing or increasing activity of circuits that are related to uh, other types of reward seeking, perhaps. And then perhaps during addiction, this circuit could be impaired in function, as has been shown for other reward related circuits. But yeah, those are all interesting future things that we haven't done.
Yeah, thank you. So there's a question here from Ariel. What mechanisms might underlie the preference for fatty and pleasurable foods like chocolate over healthier options like carrots, as observed in mice with activated VGAT PAG cells? And how might these findings inform strategies for promoting healthier eating behaviors?
So first, one thing that this work uh, kind of suggests is that a main reason for increased food seeking might be not more hunger, but kind of more anticipated craving or pleasure from eating the food. So there's this non-metabolic component of food seeking. So this work, along with other works, suggests that. And it's difficult to see at this moment how this could impact development of better therapies. But one thing that it does show clearly is, we activated only a very small number of cells in the mouse brain. It's maybe 2,000, something like that, and even that small amount of neural activity change was able to very dramatically alter the food-seeking behavior of the mouse, which shows that uh, these food-seeking disorders, they can be caused by very small changes in neural activity. So it shows that, and then it also shows that uh, these disorders can be caused by real identifiable biological events, like some cells being more or less activated. And because of that, these eating disorders should be considered a kind of real disease, which often it isn't in amongst many people. Like, so everyone considers malaria a real disease, but a lot of people might say no, eating disorder is not a real disease, just people being lazy, etc. So the fact that we can identify some clear biological circuits that cause symptoms that are seen in these conditions suggests that these are some real biological impairments in those people.
Yeah, thank you, Abby. So I saw a comment here by R about Neuralink. He said that your study might interest Neuralink. The initial findings that you have. So I just want to comment here that. Of course, I want to ask you what can you say about this, but I would just like to say that yesterday I accidentally had a chance on an interview with um, Meron Gribitz, who has a company, and I think Ed Boyden is the head of the advisory board in this
Mm-hmm.
company, and what they have is something that they are already selling to the public. I think, of course, it's FDA approved. It's about also brain computer interface, but it's not as invasive as Neuralink. It's
Mm.
just the size of a penny that you put on your forehead, and I think it also links to your physician. Um, it remotely links to your physician if he wants to try to
Mm-hmm.
check what's happening in your
Mm.
brain. And they are trying to market it. One of the strongest point that they are trying to bring about in marketing this is that they are using it as a treatment for depression. And of course, as of now, the price is like $10,000.
Mm-hmm.
So I was just thinking, how do you think could this apply to products like this? Neuralink is a little bit maybe far off, but there are products which are not invasive, which are already in the market. And what do you think are the implications in your study that would be applicable to products like this?
Yeah, I think that this brain machine interface technology is probably the most important thing that will concretely alter patients' outcomes that comes out of neuroscience in the next few years. And it's not just these things that might be important for depression, it's much wider. So, for example, there are these interfaces for people that uh, have some limited mobility due to some spinal cord injury. So I think within our lifetime, there will be some way of reading their neural impulses to make them move using some exogenous armor type thing, or people that have lost some limb, like lost an arm. I think it will be possible again within our lifetimes. There'll be patients that will have prosthetic arms that are reading the nerve signals from the rest of the arm and then making movements with the prosthetic robotic arm. So I think that kind of brain machine interface will be very important and will be seen within a few years. And like you said, all of these things will start out very expensive and clunky and inaccessible, but this technology is advancing very fast. So my guess is that this will be, cheap and small and accessible within not that many years, like within our lifespan. That will be a normal technology that you can get at your doctor. And they'll be also progressively less and less invasive as the technology improves.
Yeah. Thank you. So, one of the first guests here in Quantum Photonics when we started to invite speakers was Miguel Nicolelis, and in 2012 or 2014, he made a paraplegic who has been paralyzed for about more than 10 years. He made a paraplegic walk on the football field of
Yes, yes.
Brazil. You remember?
Yeah, I remember that.
So, yeah, so yeah, using only brain computer interface and also exoskeletons. Of course, they had the training in virtual reality for 8 months before they were able to implement that.
Mm-hmm,
And
mm-hmm.
now I'd like to inform you and everyone that the last time that Miguel was our guest speaker here, he was telling us that he'd like to be more of, giving service to the public. Maybe that is his self-actualization at this point, because I think he became chairman Duke Emerit of Duke University, became chairman emeritus of their department, and now I'd like to say that he has a clinic now in Brazil, and
Mm-hmm.
also there is a foundation and groups of people trying to help him set this up in London, and their objective is to give very cheap or free treatments to people who cannot move or who are in the ALS conditions.
Mm-hmm,
And
mm-hmm.
yeah, he's starting to do it now. So it's happening now, what you're thinking about.
Oh
Yeah, I
yeah,
just like
yeah.
to
It's already
inform
happening
you
now,
that.
but it's kind of, as you've seen, very limited and expensive and experimental, and very few patients have access to it. But this is improving really fast. Like it's already very different from what was available, say, 5 years ago.
Yeah, thank you so much, Avi. And Bob, if it's okay with you, I'd like to hand over the moderation to you if there are people who would like to speak.
Sure, I think Mustafa raised his hand. Do you have a question or a comment at this point, sir?
Yes, thank you, Bob. Actually, I have two other questions, but which are not linked to a specific study of Dr. Avi, but concern optogenetics in general.
Mm-hmm.
Can you discuss cases where optogenetics has directly contributed to breakthrough in understanding or treating specific neurological disorders?
Uh, yeah, there was a case where it gave back some light sensitivity to a particular type of blindness, which was using a real. patients. That's something that happened. And then there's also plans to use it eventually for more, not brain, but peripheral nervous system problems. So, for example, this hasn't happened yet, but these are more in development possibilities. Like, say, if you have chronic back pain, which is often due to some pinching of some nerve or something like that, because it's in the periphery, not in the head, it would be easier to like optogenetically inhibit that to decrease that kind of pain. But this increasing of light sensitivity to in a particular type of blindness is something that has happened already with some small number of patients.
Great. My last question, what is the potential for integrating optogenetics into regular clinic therapies? And
So
what
sorry,
are
integrating optogenetics with what?
into regular clinical therapies?
Ah,
And what steps are necessary to move from laboratory research to clinical applications?
mm-hmm. uh, so as someone already asked before, I think it seems not that easy or likely that optogenetics will be used in some large scale to treat millions of patients because it's very invasive and you have to have a laser light source the person is walking around with to use a laser to activate the cells, and it requires a virus injection. So there's both this laser and the virus are two big problems to solve. But the insights used from that are gained from optogenetics can be used to guide the development of therapies, because if we find that this particular group of cells when active causes some bad symptom, then essentially any discovery that any other method that allows you to control the activity of those cells could be used as a therapy, which could be some kind of drug that you take orally. Maybe that drug alters the activity of those cells, and then the behavioral outcome will happen even without using optogenetics in the patient. So you could, so the value is more in the learning which brain circuits control which symptoms, and then if there is some secondary discovery of how to control that circuit not using optogenetics, that would also have this a therapeutical benefit, which could be both through some drugs that you can take orally as a pill, or could be through some kind of intervention like exercising more or doing meditation or eating this kind of thing, or anything that essentially alters the activity of those cells discovered optogenetically would also uh, change the behavior, presumably.
Thank you very much, Dr. Avi, and good luck for the rest of your research.
Oh, thank you.
Research, you're
Thank you
very,
for the
very good.
insightful questions.
Yes. So I think there's a question here from Mirna on the room chat.
Mhm.
I don't know if you've answered this. So she is asking, may I ask Abby this question? What portion of the brain governs panic as you described? Maybe she came
Uh,
in late.
yeah, so the part of the many different regions have been found to control aspects of panic, but the region most strongly implicated in panic is this region we studied here, which is the periaqueductal gray region. And basically, if you excite all the cells in this region, it causes these panic behaviors. But here we didn't excite all the cells in this region. We excited and activated only the GABA neurotransmitter releasing cells. And then that more specific manipulation did not cause panic and instead caused more of this food seeking types actions.
Yeah, thank you, Avi. So we've been here for about, I think, I don't know if I saw LT flashing her mic. Uh,
Uh,
I just, yeah, LT, let's go.
you ask whether there's a time for me to ask questions. You check with Ivy first. Go ahead. Or shall I ask
Uh,
question?
yeah, go ahead, please, LT.
Okay. My question is that I mean, how is your assessment? You know, you've inserted this ion channel, right? It's because you add in, you inserted this. Would that alter the function of those neurons who express this ion channels?
Sorry, could you say again? Would what alter the function?
Because this particular region you focus on, roughly you said that there's GABAergic neurons, right?
Yes.
And so because you infect them with this virus, which would be that's how it responds to your light on, like on and off switch. So those neurons presumably you don't know how, I don't know how many ion channels they inserted, you know, like whether those insertions, so it's like extra, correct?
Yeah, those channel rhodopsin proteins were extra and made by the virus infection, so they don't exist naturally. So it's something in the order of many thousands of these
Yes,
extra channels
that
in each neuron.
that yeah. So my question is that would that have any effect on its
Ah,
function? You know, that's my question.
oh, okay, I see. Yeah, it's a good question. So this we can answer. So it appears that no, because the effects we see don't happen just after the virus infection. So they only happen when we also deliver this laser blue light to activate the channels. So in the
So,
absence of the blue light through
mhm.
the laser source, the mice with the virus infection don't seem to do anything measurably different from the control mouse.
From the control mouse, and with how many cells we're talking about those GABAergic neurons in this particular region?
Well, we didn't count, but it's not some enormous number. It's probably
Okay.
something around 5,000 or 5 to 10,000 cells
Uh-huh.
at most. So might be something between 2 to 10,000. Yeah, it's
Two to
not
10.
fewer than 1,000, and it's not something like 50,000 either.
Okay. And on your slides 13 or 12, and you said there's those each those like lines, right, showing from a single. So you were doing the single neuron recording then?
Yes.
Okay. So you have like about a dozen, maybe 20, around 20 of them. You specifically, those are the really identifiable single neuron recording, correct? That's just I wanted to confirm.
Yeah. Each line there is one cell,
One cell.
and
Okay,
then at the
thanks.
same time we can record
The
something like
group, the
100 cells.
so it's like average. So you have both. You have done this single neuron recording versus you have done this whole like hundreds of together, average them out, right?
No, no,
Activity wise.
not average. We recorded 100 single cells
Oh,
at the
okay.
same time,
At the
so we
same
can
time,
see what the 100 different cells, around 100,
around
approximately per
100.
mouse.
Oh, okay, okay, okay. Thank you.
Yeah.
No more questions for me. Thank you
Okay.
so much.
Thank you.
Yeah, thank you, LT. And Kevin also flashed his mic. So, yeah, Kevin.
Oh, yeah, I got a future experiment maybe. Stimulate sexual. Well, that's all I can think of for addictive for a mouse. Their sexual, and compare that and see if it disrupts. This you're finding. And then I'm sure the government would love to you get funding for that. And basically you can tell people that if they're related, your urges are just the same, for either way. So you're just being controlled by your urges. If those two are related, is all I
Sorry.
can think of. Or the only other, the four Fs. Food, flight, fight. So that's all I can think of for that.
Sorry, that's if which two urges are related?
All I can think of is like the four Fs. The joke.
Uh huh. Yeah.
Flight, fear. Fight or flight.
Uh huh.
Food and sex. So I'm thinking sex. So if you stimulate the neuron for sex, put a partner there, even if they're not in the mood or the correct partner. Just and it stimulates them instead. And actually, it's all related. Because porn has been the main Google. People that's the main result for Google since the inception of the internet by
Mm-hmm.
far.
Mm-hmm.
Porn is the main search.
Mm-hmm.
So yes, so if those two are related, you can do a public announcement, public service announcement saying that if you can control your urges for fast food, for junk food, because it's the same thing as sex, porn, food porn they call it,
I see.
so you can do maybe some type of message saying, you know a gut message, saying you guys are you have to control your urges. It's the same thing.
Yeah, so that we also tested, and then yeah, these cells don't seem to increase or decrease any mating in mice. So it seems not related to that urge
Oh yeah,
in
but I mean,
this circuit. But there could be other circuits that control both.
oh
But
yeah,
this
yeah.
one
That's
does not.
oh okay. Yeah, I don't. Yeah, I don't know. I don't know if there's other ones. Yeah, that's what I mean.
Uh, Thank you, Kevin. So we've been here for 2 hours now, and I think I will ask Avi the last question.
Sure.
So, yeah, Avi, is there a follow-up study that is upcoming? I know that it's not very easy, maybe, to get some funding for the studies that you have. So, but I'd like to ask because it's very relevant.
Mm-hmm. Yeah, we are already following up on this study. So now we are uh, studying more detail what foods, when activated, does it cause eating more or less of. We're also expanding to try to see if it has role in drug-seeking behaviors like addictive drugs and also other ingestive actions like drinking water. So we're trying to expand and see what more this controls.
Yeah, thank you so much. Please let us know if you have your next paper. We would be very eager to listen and learn from what you had known in your study. So, yeah, at this point, I'd like to thank our guest speaker, Dr. Avi. And yeah, please come back here in Clubhouse anytime. And I would also like to thank everyone who had been listening to us and supporting us. And yeah, if Darcel is here, so I'd like to ask Bob if he has some last comments before I hand over the mic to Darcel. And of course, I'd like to ask Avi if he has a message for us.
Uh, yeah, my last words would just be that although it might appear esoteric to study how the brain controls something in mice, uh, in the long term, these sorts of scientific discoveries are the basis for new improvements in quality of people's lives, right? So, for example, now electricity is important for every human activity we imagine. But when they were first studying electricity in the 70s, in the 1700s, in 1750, no one knew it's going to be that important. But now it is. So, and if they hadn't done those basic science studies, then we wouldn't have all of this new technology now. So it's very difficult to predict which thing will become very important in the future. But it's very easy to predict that whatever is important in the future, that's a new technology, will be due to some scientific discovery that's happening now in the present. So um, that's why I urge everyone to try to be interested in these and I know all sorts of other scientific developments.
Yeah, just please know that we are rooting for you and your researches. I think just a few hours ago, I read some interview with Eric Schmidt because he is also supporting a lot of studies, and he said that we have to prepare for researchers in bio and also neuroscience to be exponentially moving because of LLMs and AI. So, can you say something about this?
So AI, yeah. So I think AI will, make some jobs become obsolete, but it'll also create some other opportunities that we don't understand at this moment. And we use it in our lab for kind of machine vision related tasks of finding behaviors in mice, and we also use it a little in writing software code to analyze data. So I think it will become a more and more important tool in scientific discovery.
Yeah, thank you, Avi. So I'd like to give the mic now to Bob to wrap it up, and finally to Darcel to help us close the room. Thank you, Kevin and LT, who are still on stage, and for everyone who came on stage earlier, and everybody who has been listening to us: Lisa, Ash, Kautar, Sel, Shane, Pin, V, Mirna, Jocelyn, Kurt, Karen, Ivan, Roy, and R. So yeah, thank you so much. Mike to you, Bob.
I found this fascinating, Doctor, and on behalf of everybody in the audience, I wanted to say thank you for a great presentation. And
Well, thank you for giving the opportunity to share this work.
so I think at this point we'll go to Darcel. Are you there?
I am. And thank you, Doctor, for a great talk. To close the room, we're going to start our countdown from lucky number 6. Is everybody ready?
Ready?
Ready.
Yes.
6,
Six.
Six.
LT 5,
5.
Kevin 4,
Quattro.
Bob Thrice, Bob 3,
3 it is.
Cecile Dos Eques.
Dos.
Good Doctor, your numero uno. Call out our last number, please. Doctor?
Abby, maybe you forgot to
One.
turn on your mic. All right. So thank you so much, everybody, and thank you, Dr. Avi. Uh, Thank you so much, Avi. We will say goodnight to everyone, and thank you for participating and patiently listening. We will see you in the next rooms.